Hello and welcome back. Now that we have gone through the mechanisms of how all the solutes filtered at the glomerulus are reabsorbed and sometimes secreted in and out of the nephron, let's go through specifically sodium and potassium and look at the factors that regulate the reabsorption or secretion of those two solutes along the way. First, we're going to start with the factors that stimulate the reabsorption and secretion of sodium. Before I get started and going through these steps, I think it is important to highlight that the logical kind of sequence here. If you have low body sodium, you might think to yourself, well, if we have low sodium, then we just need to hang on to as much sodium as possible. But in reality, when the kidneys are involved, a sodium problem is usually a water problem. Okay, so let's walk through this and see if it makes sense. Because in order to regulate sodium, water has to be involved. So when we say that we have low serum sodium, is it because there is a true lack of sodium molecules in the body? For instance, what is the number of millimole of sodium? Is the number of millimoles of sodium too low, or is it the fact that you actually have normal sodium, but the concentration is low because you have a water issue, you have too much water? And typically, it's not necessarily the fact that you don't have enough individual sodium ions in your body. It's that you have too much water that they are dissolved into, which reduces the serum concentration. These are all factors that can contribute to a decreased serum sodium. And as a result, the kidneys are going to want to compensate by increasing. So you're going to compensate by increasing sodium. Similarly, that means that there's two ways you can go about doing this. You can increase the number of molecules, or you can change, that's what my little delta here is, the concentration of sodium. And therefore, over here on the left in this graphic, when we look at a salt deficit, this is a stand-in for low concentration of serum sodium. When you have a salt deficit, the kidneys and the body are going to respond by changing your overall water balance. That is why, in this case, if you have low serum sodium, the response is a low ADH, so drop ADH. When antidiuretic hormone levels drop, that means you're going to diurese a little bit. As you diurese, you're going to be eliminating water. Eliminate water. If you eliminate water, but hang on to your serum sodium, what's going to happen? Okay, so as a result, your blood volume goes down. As a result of the blood volume going down, your effective circulating volume goes down. That's going to activate the renin, angiotensin, aldosterone system. Aldosterone will serve to hang on to sodium. So there's two things that are going to happen at the same time. One, you diurese water, which is up here. So this is the first thing, and then eventually you get to this one, which is the renin, angiotensin, aldosterone system. And that will feed back into this process of the action of aldosterone promoting sodium reabsorption will help you hang on to the actual number of molecules of sodium, millimoles of sodium, but the low ADH is going to result in a water change. That water change will only serve to increase your serum sodium concentration through both ways. If you look at the urine of this individual, That's what this kind of flask or Lermeyer flask is representing, and all these dots are supposed to represent sodium. So the urine of this individual would be a hypotonic urine. Okay. Now go back to the most important factors that stimulate sodium secretion. So go back up. Our rule here is usually when we have a sodium problem, it's usually a water problem. So when we have a salt excess, or you could say an increase in serum sodium concentration, that's usually going to be affecting water. So the brain is going to sense the increase in osmolality. We raise ADH. We have a water retention because we're in a state of antidiuresis. That raises blood pressure. It also raises effective circulating volume. That effective circulating volume raise will do a couple of things. One, it is going to serve to stimulate ANP. ANP will then go on and stimulate the process by which sodium can get secreted, or in this case, a drop in reabsorption. That ANP can go on and inhibit aldosterone. That's what this arrowhead with the line next to it is. Similarly, the raise in blood volume and blood pressure will stop ongoing renin-angiotensin-aldosterone. So I'm going to put a little line through that so that that doesn't go on. Because in this case, we had too much salt, so we don't want aldosterone. We want to eliminate sodium in the urine. So in this case, we're in a state of antidiuresis, so we're hanging on to our body water, and we are eliminating sodium in the urine. So this will be a hypertonic urine as a result. Now that we have gone through the regulation of sodium, let's turn and focus on potassium now. The most important factors that stimulate potassium secretion are written here in the box, and they are increased serum concentration of potassium and aldosterone. As opposed to sodium, where usually in the case of sodium, sodium concentrations are usually related to a water problem. That's not necessarily the case with potassium, and the reason why is there is just simply so much more sodium in the extracellular compartment than there is potassium. And so as a result, when we say increased serum concentration of potassium, we really are focused specifically on the number of potassium in terms of millimoles that there is in the extracellular compartment. And then aldosterone is an important factor of potassium secretion. When it comes to aldosterone, aldosterone does a couple of major things, but aldosterone, you will be well positioned to remember that it increases sodium reabsorption and it increases potassium secretion. And that is because of the relationship in that exchange between sodium and potassium. Over here is a reminder once again that potassium reabsorption is most likely to take place in the proximal tubule. Some takes place in the thick ascending limb. But then when it comes to secretion, secretion takes place in the collecting duct, okay? So secretion can only take place here. Everywhere else is reabsorption. Continuing and focusing on potassium, let's now focus on the reabsorption of potassium. The major factors that stimulate potassium reabsorption are similar to the factors that stimulate sodium reabsorption, only rather than it really being a water problem, it really is about potassium. So if you're low in potassium and for any given reason, you're going to want to hang on to as many potassium molecules as possible. And over here is again a reminder, the places that you will most readily reabsorb potassium is in the proximal tubule, where you have 2/3 or 67% of all potassium reabsorption takes place. Thick ascending limb, if you remember the thick ascending limb, the way that potassium is reabsorbed is through the NK CC2 channel. And then in the collecting duct can reabsorb potassium, especially through these alpha intercalated cells in the process that we went through earlier in this slide series. So that's through the alpha intercalated cells. So depending on the needs of the body at any given time, you're probably always going to be reabsorbing some potassium in the proximal tubule and thick ascending limb, but secretion or reabsorption in the cortical collecting duct is dependent on the needs of the body at that time. Continuing on with our theme of regulating transport in the nephron, this section with Learning Objective 3 is focusing on ADH. I know that this is not your first introduction to ADH, so this is just a brief review of antidiuretic hormone. It is also called AVP and vasopressin, just in case you come across those terms in different resources. And there are two major things that will stimulate ADH release, and they are hyperosmolality and volume depletion. Of these two things, it is the hyperosmolality that is more sensitive to the release of ADH than volume depletion. And that's because if you think about it, osmolality is very important in terms of maintaining electrical conductivity in the brain and the heart and all these areas, whereas volume depletion, you can get by being dehydrated for a while. But if you have a change in osmolality, that can have some pretty big impacts on the body. Therefore, we say the ADH is more sensitive to small changes in osmolality, or I wrote osmolarity here, but it's osmolality. It's more sensitive to that than it is to similar percent changes in blood volume. And this graph is depicting that. If you have a percent change down here on the x-axis, so if you, that's what is shown down here, this is the subsequent release of plasma levels of ADH. So if you have, let's say, a 5% increase in ADH, or sorry, if you have a 5% increase in either one of these two things, either volume or tonicity, the tonicity is shown here in these black dots. So these are the volume and isotonic volume change. So meaning you're 5% dehydrated is kind of what that is saying. So you have a drop of 5% of your isotonic volume. That's what these black dots represent. Then you get a ADH concentration in the serum of about, I don't know, 3 picograms per mil. However, a 5% change in osmolality will result in, 12 picograms per mil of ADH, meaning that at the same change of percent, 5% osmolality increase will result in 4 times the amount of ADH. So what does that look like? Well, let's say you have a normal serum concentration of, and I'm just going to make something up here. If you have 200 milliosmoles per liter, a 5% change would take you up to 210 milliosmoles per liter. Okay, so that's a 5% change. So as you can see, that doesn't look like a big difference. You go from 200 to 210. That's only an increase of 10 milliosmoles, but that is huge for the body's electrical conductivity. And so as a result, you have, in this case, you go from 3 to 12 picogram levels of ADH in the serum. So it's a big increase. So it's much more sensitive to osmolality than it is to volume. As an aside, and from a nutritional standpoint, this is very fascinating to me. The recommended minimum intake of sodium is 500 milligrams of sodium per day. So if you take in 500 milligrams, that's half a gram of sodium, then in your diet, keep in mind the average diet is taking in 4 to 5 grams per day in this country. Equate that to a teaspoon of table salt, which is, sodium chloride. A teaspoon of table salt contains approximately 6 grams of sodium chloride, or 2.3 milligrams of sodium. So if the minimum intake is 500 milligrams of sodium per day, a teaspoon of table salt contains 2.3 milligrams of table salt, and the average person is taking in 4 to 5 grams per day. That is a lot of salt. So from a nutritional standpoint, maintaining proper changes in or proper serum osmolality is hugely important, and there is a big nutritional impact. So now that we've reviewed ADH, let's go into what I call the big important things that ADH does, the 3 ADH biggies. When learning about ADH, the most important and famous mechanism that everyone focuses on is this one, number 1, and that is that it increases water permeability in the principal cells of the latest part of the distal tubule and collecting ducts. It does so through the insertion of aquaporin channels. And we talked a little bit about aquaporin already and how principal cells can have insertion or changes of aquaporin. So this is the most famous mechanism. But there are 2 equally important mechanisms that ADH does. It increases permeability of urea in the inner medullary collecting duct, so inner medullary collecting duct, but not in the cortical or outer medullary collecting duct. Okay, so the inner medullary collecting duct, so the way far down in the bottom of the nephron. It also increases activity of the NKCC2 co-transporter in the thick ascending limb. So if you remember the NKCC2, then it is responsible for reabsorption of sodium, potassium, and 2 chlorides. We're going to go over each of these in the next couple of slides and in turn and focus on why that's important and why ADH does this. First up is that first function of ADH, of the 3 ADH biggies, and that it is increasing water permeability in the principal cells. And it does so through the insertion of aquaporin 2 channels or pores in the apical membrane of those principal cells. This graphic on the right is depicting what essentially is happening here. We have the urine side on the right with this yellow tube representing the nephron. I'm missing in there. And then the blood side is over here on the left. So this is our blood capillary. And these are representing our principal cells. So in the presence of ADH, you have fluid, and I have these little water droplets to represent water flowing through the nephron. ADH will literally take these aquaporin channels and stimulate a signaling cascade that results in their insertion into the plasma membrane of the apical border, which is right over here, the apical border of these principal cells. So these aquaporin 2 channels right here get inserted into the membrane. That allows for water to then flow through the aquaporin 2 channels. The water can get inside the cell. And then how does water get out of the cell? Well, it gets out of the cell through aquaporin 3 channels, which are always in the basolateral membrane. So these are always around. They don't really change or do anything. The aquaporin 2 pores, or those channels, are variable. So they will only show up when ADH is around. In the absence of ADH, without ADH, those aquaporin 2 channels, they stay in the intracellular compartment. So you saw on the last slide, those aquaporin 2 channels were inserted into the apical membrane of the principal cell. Well, this is basolateral on the left. Now, in the absence of ADH, they just stay inside, and they're not doing anybody any good inside the cell. Water can't get in. There's a barrier here. That's why I have my little meme over here from Lord of the Rings, if you remember this, saying the way is shut. Water can't get in. So if water can't get into the cell to get reabsorbed into the bloodstream, where does it go? Well, the water can only go one place, and that is on down through the tubule towards you know where, and that is the bladder. So in the absence of ADH, we are in a state of diuresis, and that is water being excreted. Okay? Now, hopefully by this point, you're starting to get a sense of understanding a little bit of what's going on, having a good sense of how sodium and potassium and all those things are regulated, and now you've got a good grasp on how water gets reabsorbed. Well, now we're going to throw in a new player in this game, and that is urea. Okay, urea, I'm going to go into more detail later on when it comes to regulating urine concentration and urine dilution. But for now, I'm going to introduce urea as being involved as with ADH. So there's a concept called urea recycling, and this only occurs in antidiuresis. So whenever we have the presence of ADH, not only do we have those aquaporin 2 channels being inserted into the principal cell apical membrane, now we're going to stimulate this process called urea recycling. So let's look at this graphic here. This graphic is showing the presence of ADH, which you know because water is getting pulled out of the nephron. Okay, so water is getting pulled out of the nephron, meaning there's not going to be much water. Not much water is left to go to the bladder. Okay, so we don't have a lot of urine being built up here. Now, because water is being reabsorbed in the cortical collecting duct, urea gets kind of left behind. So all these water molecules, I'm going to put little water molecules, they're getting sucked out, okay? And that means that anything that's left behind in here is getting more and more and more concentrated, such that by the time you get to the lowest most portion of this inner medullary collecting duct here, so we're in the inner medulla here, the outer medullary collecting duct would be about here, and then the cortical collecting duct is up here in the cortex. Urea gets really, really concentrated because all the water has been sucked out, but not urea, so urea gets concentrated. As a result, urea, by the time we get to the inner medullary collecting duct, is so concentrated that it moves down its concentration gradient. And this is a passive process. Urea moves out of the intermedullary collecting duct and is floating around now in the interstitium. So this is the interstitium, okay? And we'll come back to this in a little bit. But essentially what happens to urea is it gets pulled back into the loop of Henle, and then it just starts over again. It just goes back through the whole system and then cycles around, and it makes this huge cycle over and over and over again. So, second part of ADH is the stimulation of urea recycling. And then the third big thing that ADH does is it stimulates the activity of the NKCC2 channel in the thick ascending limb cells. If you remember NKCC2, it is responsible for reabsorbing sodium, potassium, and chloride. So ADH not only results in the insertion of aquaporin channels in the principal cells, and it is involved in urea recycling. It is also going to stimulate reabsorption of sodium, potassium, and chloride in the thick ascending limb. And as a result of the back leak of potassium through those ROMK channels, if you remember, potassium gets pulled inside the cell, and then it can either back leak back out into the tubular lumen, or it can get reabsorbed through potassium channels. That builds up a positive charge here. That positive charge repels magnesium and calcium, which get repelled and passively reabsorbed through the paracellular pathway. Therefore, ADH has an indirect effect of increasing reabsorption of magnesium and calcium as well.